So hi everybody, this is Mihaly Thomas, microbiome lead for DNA Nexus, and welcome to today's webinar. Today we're going to talk about the Mosaic community challenges, specifically strains 1 and the winners of this challenge. We're happy to have representatives from both participations presenting today. However, before starting, I will start by giving a review of the current ongoing challenges that we have on Mosaic right now, and then Rayan Chikhi from CNRS in France will present his mini assembler, and CosmosID will present us their work. So as I said, my name is Mihaly. The moderator for today's webinar is Brenton. If you have any questions, you will see on your Zoom window that there is a couple of options to ask the questions. One is to raise your hand, at which point we can give you access to your microphone so you can ask your question. Or if you prefer to just type your question, you can click on Q&A to type your question, and Brenton will ask it to the other participants. The organization of this raise hand and QA icons might be different for your browser, so please keep that in mind. So just a quick reminder about Mosaic and what we're trying to do with Mosaic. Our mission is to provide a cloud platform that enables the microbiome research community to accelerate the improvement of methods through unbiased benchmarking. And we hope to achieve this unbiased benchmarking by administrating community challenges where everybody and anybody can participate, and they can use whatever approach they want to use to address the questions we pose. So far, we have launched 3 challenges, strains 1, which finished a couple of months ago, and this is what we're going to discuss today in detail. However, right now, we're having clinical strain detection challenge that is running, and also recently we launched the standards challenge. So before I pass the podium to Ryan, I will discuss a little bit about these 2 challenges and give you some details that you might need to know. So the clinical strain detection challenge has a goal to benchmark the ability of metagenomic tools to detect the presence of certain known strains in a sample. So with this challenge, we're trying to address a very fundamental question, which is basically, can you tell us, if we give you the reference genome of a bacterium, whether it's present in a microbiome, if we give you the metagenomic sequence of that microbiome? And I think everybody can appreciate the importance of this, or the ability to answer this question accurately. One example why this is important is to be able to assert whether in a biotherapeutic approach, whether the strains that you're giving to the patient are actually engrafting in the patient, are they washing out, and also whether some other species or strains are being augmented, basically changing their behaviors. Another obvious application of this capability to accurately detect strains, of course, is diagnostics. What we're trying to do with the clinical strain detection is simulate the case where you have 40 isolate genomes that are you giving to a patient, and then you want to be able to see whether that patient has those strains, right? And the way we constructed this challenge is to isolate bacteria from real stool samples from different humans, sequence the bacteria, and also sequence the microbiome samples. And this is what we give to you, the user or the participant. We give you 40 isolate strain genomes and also 4 metagenome sequences. And what we ask you to do is basically identify the source of each isolate bacterium. And we know the answers because we did the isolation. And actually, when we say we, I mean Jeremiah Faith at Mount Sinai in collaboration with the Janssen Human Microbiome Institute, with the sponsors of this challenge. So it's pretty straightforward. Like the previous challenge, we give you 2 sets of data sets. One is the training data set, where you get access to the 4 metagenomes and also the 40 isolate genomes, the sequences of those genomes. And also we give you access to the truth table. So we tell you exactly what the answer should be. And the purpose of this is for you to be able to optimize your tools and play around and see how this challenge works. Of course, for the challenge part, we only give you the 4 metagenomes, which all of them are newly sequenced, so they're not available in public anywhere else. and also 40 genome sets. Of course, because this is a challenge part, we only give you the truth after the end of the challenge. The format for the sequencing reads, of course, is FATQ and FATday for the genomes, and the truth tables are in tab delimited text files. The way you're supposed to submit your analysis is using these formats that we provide for you. First, there's two options. First, one you can just provide as a presence absent file, where 0 means the strain is not present in that specific sample, and 1 means that the strain is present in sample 2, for example, for Alice type T3. The other option is to provide a float number, which is a more probabilistic approach. This float number will indicate the probability that the strain is from a particular sample. This allows us to calculate a precision recall curve for the submission. However, this precision recall curve is not used in the evaluation. This is just for us to contain, to have rich data in order to do the comparisons and provide to the community. So for this challenge, we know that a lot of our participants are spending a lot of time to analyze them. So we want to make their time valuable. So for the winners, we're going to give 3 prizes. For the first prize, we're going to give $10,000 to the winner. The runner-up gets $5,000, and the third place gets $2,000. So the challenge closes on July 25th. So please participate. You have enough time, and this should be a great challenge to evaluate your tool. So before we go to the next challenge, I will quickly give you an overview of how you can participate in this challenge. If you go to mosaicbiome.com, you will be able to see the challenges in the challenge tab and click on the Explore Clinical Strain Detection Challenge in order to participate. Of course, I have already joined. If you haven't, you'll be able to join, and when you do that, you will get access to the testing, to the assets for the participation to this challenge. When you're ready to submit, you can come to the testing ground to submit the analysis of the testing data by clicking Testing Ground Submission. And of course, you'll be able to see other users' participation. Now, one thing I would like to note here is that all these users have access to the truth. So if the scores seem very high, please don't be discouraged. They might be trying different things about the user experience on our platform. When you're ready to submit to the challenge, you can go to the Challenge Submission tab on the left, and you can submit the results for evaluation here. In this case, you will not be able, however, to see the results until, of course, until after the end of the challenge. So the second challenge that we have running right now is the Standards Challenge. And this challenge aims to capture the diversity of protocols for NGS-based microbiome profiling methods. And of course, I think everybody who is involved in microbiome analytics appreciates the need for standardization of protocols. So of course, that is needed for accurate and producible microbiome profiling analysis. And there's many places in an experimental protocol where variability might be introduced. For example, anywhere from sample collection and storage to nucleotide extraction and so on. So the aim of this challenge is to, at the first level, the high level, to capture diversity of protocols. At the second level, we would like to also see what is the effect of these choices in experimental protocols on the interpretation of the results. So we are excited for this challenge because it combines both the experimental world and the bioinformatics world. What we have done is collaborate with the BioCollective, DNA Genotech, and the Janssen Human Microbiome Institute to create 700 kits that are available to ship for free to anybody who requests them. These kits contain 5 real stool samples. And also, we're excited to have Scott Jackson from NIST participate as well as a collaborator, and he's providing us with 2 mixtures of DNA extracted from bacteria at known stoichiometries. So if you order these kits, you'll get 7 tubes that contain these samples. What we expect you to do is sequence these data sets and then come back to Mosaic and upload the results, both of the sequencing, but also the metadata that you use to sequence these samples. When you do that, our platform will automatically analyze them and present those results to the rest of the community in an on-demand interactive way. So our users or all participants will be able to see both the results of the analysis of this data set, but also be able to see statistics about the different protocol choices made by different labs. I should mention that, again, Mosaic is a pre-competitive space, and we want open participation. All the data and metadata collected by this challenge, and all challenges, in fact, will be made available for download by the users, and you will be able to analyze the data using your own approaches, and if you want to publish them, you'll be free to do so. Going back to the platform to show how this works. Right now, you can only order samples, and to do that, you have to go to the standards challenge, and if you click request samples, you'll be presented with a form here where you can give us your name, address, and other details so that the BioCollective can send the samples to you. I should note here that you may order up to 3 sample kits. And if you want to order more than that, please let us know, and we can help you request more. All right, so the main purpose of today's webinar is to discuss the results for strains 1. And just a reminder, for the first challenge, what we wanted to evaluate is different bioinformatic methods that are used in the analysis of microbiomes. And what we present today is the results for profiling and assembly. What we provided to the participants is 4 data sets, 1 real and 3 simulated. And again, one of the strengths of this challenge on Mosaic is that we provide real biological data sets where the truth is known in order to evaluate the performance of different tools. Again, another strength for our platform is that your participation is immediate. You get feedback immediately, and also we provide interactive visualizations to navigate the results. So for the assembly part, Rayan Chikhi performed the best in the cumulative genome fraction score. So congratulations, Rayan. But we also have to mention a couple of other submissions that performed pretty well. One is Peter McCaffrey from Texas Medical Center with his submission Deep Biome, whose submission had the best compromise between the genome fraction percentage and also config size. And also in terms of the length of assemblies, Sergey Nerk with his Metasphere assembler achieved the highest length of assemblies. I should also just mention here that we made some choices as Mosaic developers to focus on specific metrics. There is no, when we were setting up this challenge, it was very difficult trying to decide how to evaluate it because there's so many different ways that the community uses assembly. And what we decided in the end is to just evaluate the genome fraction. So in order to view the results of this challenge, you can go to challenges, and you can explore the Mosaic Community Challenge Strains 1. Once you do that, you can visit the challenge submissions, and you have the option to explore profiling and explore assembly. If you click explore assembly, you'll see all the submissions. We have 9 submissions for the assembly part of Strains 1, and I think that answers the question of one of the participants of the webinar today. But also here you can see very high level statistics for the different submissions. You can see that Ryan's submission performed the best across all metrics that we're looking at. And additionally, we provide options to dig into these data sets. For example, you can perform comparisons between the different data sets for the different metrics that we provide. For example, you can select mismatches, and it will automatically tell you how each submission performed. Additionally, you can perform pairwise comparisons between different submissions. If I compare, let's say, Ryan's submission with Sergey's submission, I can see how they differ in terms of genome fraction. For example, in this case, we can see that Rayan performed better in this specific bacterium, right? So I will provide that rich data source here for you to investigate. Additionally, going back to the submissions, you can get many more details if you click on the submission itself. So you can see exactly how each submission performs. We also provide an interactive meta-class report. All right, so I will not belabor this point. I will turn the podium to Rayan. And again, if you have any questions, please raise your hand or ask them through the chat function of Zoom. So.
All right, testing.
So, hey, Rayan. So I think you have control now.
Let's see. Yes, I do. Okay, should I start?
Yes, go ahead. Sorry.
Yeah, sure. Well, thanks, Mihalis, and everyone at Mosaic Microbiome for organizing these challenges and also for giving me the opportunity to speak about it today. So I will talk to you for about 10 minutes about the assembly entry at this challenge using a mini assembler. And so I mentioned that those slides are available on my webpage if you want to see them later. I just briefly recall the problem of metagenome assembly. So you probably are aware of it, but the way I would define it is to reconstruct a set of genomes from species and possibly go as far as strains. That was the purpose of this challenge, from typically short-read sequencing data, so Illumina data, from an environmental sample. For this task, there are many challenges, which are at least 5 of them from the space presentation. And the most challenging task would be to reconstruct closely related strains. So that was exactly the purpose of this challenge. But there are also other difficulties of metagenome samples, such as low depth, uneven depth, speed, repeats, their large sizes. In the challenge, there was 3 different data sets ranging from low complexity, small size, to high complexity, large size. And also note that mostly nowadays metagenome sequencing is still short reads, but maybe long reads will come later. There's multiple ways to do metagenome assembly, and I've listed a few software with the 3 most famous ones in bold, and the one I will talk about today is Minia Pipeline. And just a brief 1,000 feet overview of what a metagenome assembler does, which applies to most of the software I've shown in the previous slide. So the first step is to construct a graph from the reads. Second step is to remove sequencing errors from this graph. And the first step is a critical one. It's where SNPs and differences between repetitions and also strains are removed from the graph. So if you want to assemble strains, it's probably a good idea to look at this step specifically. And in fact, in the Minia submission for this challenge, I decided to skip step 3 entirely. And finally, step 4 would be to return contigs. So basically the result of your assembly is all the simple paths in the graph. So in this figure, it shows 3 contigs, 1, 2, and 3. And assemblers have many more steps. I've put extra steps, but in fact, it's many important ones, such as repeat resolving, scaffolding, and those steps are not performed in Minia, and we didn't perform them in this submission, yet I still note that they are very important. That's an overview of the Minia pipeline. So it starts on the top from the reads. There's error correction, which didn't perform in this challenge. And finally, the big box is a multiple K framework for assembly consisting of 3 steps. The first step is K-mer counting, done using the DSK software. Finally, after that, we do Unix assembly using bcalm, which produces a FASTA file already. And we go from Unix to contigs by doing simplifications such as tip removal, and that is performed in the Minia software. And I will make a note that all these 3 steps are actually implemented in a single software called Minia, but I wanted to separate those different steps because there actually exist different software for performing them. And the result of all of that is a set of contigs which are the assembly, and there's also possibility to perform scaffolding. And we use the best genomic scaffolder for that. It's not a metagenome assembler, so we did not actually run it for the Mosaic challenge. So we just used contigs. I will also make a note that there's a useful technique for metagenome assembly and also genome assembly called multiple K. And it's very easy to describe in this schematic. So you start from input reads and you run the assembler with a small K value. And the result of the assembler is given again to the assembler with a higher K value as well as the reads. And you keep doing that with increasing K values. I've just shown 3 here, but in fact we can go many more K values, and the result of the last step is the final assembly. Doing this enables to increase contiguity and general quality of your assembly, genome fraction also. And this is what we did also, and many of the popular metagenomic assemblers are doing this framework. Now I'll talk a little bit about the Mosaic challenge, our strategy. So there's 3 aspects. And the good is that we performed multiple K assembly with conservative simplifications. By that I mean we didn't do any budge removal, bubble removal, and other type of simplification of the graph. We just try to remove the errors and that's it. So that leads to high genome fraction because we essentially do not remove most of the k-mers, and also limited number of misassemblies because we do not make choices that lead to misassemblies. The bad is that we did not perform bubble removal, which is a typical assembly step, and it leads to a larger than expected assembly. So biologically, this has some issues. So sure, you have most of your strains present in the assembly, but it's not clear what contig corresponds to what strain. And you may also have short contigs because bubble removal not only removes k-mers, but increases the size of contigs. And finally, the ugly is that we forced QUAST, which is the evaluation method, to consider all contigs, not just the one larger than 500 base pairs by default. And we did this by just adding ends to make them larger than 500 base pairs. So as a result, we get a higher reported genome fraction than competitors which did not or might not have done this step. And I would argue that performing this step is actually forced of contigs before considering contig. The point here is that even short contigs can be very informative for the microbiome analysis, especially for strains. Okay, in this slide, I'm showing some data that I collected while performing the training data set at the Mosaic challenge. So I ran 3 different methods derived from Minia. The first one is just computing unit ticks using the BCALM software without any graph simplifications. So we have a nice N50 already 100 kb, a nice genome fraction, most of the genomes, most of the strains are reconstructed in the assembly, and low number of misassemblies. And you can see that if you go from Unitix to the Minia pipeline, so more advanced processing with just tip clipping, which is what we actually submitted to the challenge, there's a higher N50 and more or less the same genome fraction and some more misassemblies. And if you run the whole assembler, well, you will also increase the N50, which is actually, I didn't mention it, but N50 is a measure of average length of your contigs, some contiguity measure. You want to have it as high as possible, but it wasn't evaluated by Mosaic. So bottom line is that those assemblies in a low training data set are more or less comparable, except that some are longer than others. And also some remarks on the slide, I won't have time to mention them. Going from the low data set to the high training data set, also applying the same free software, we see some very significant differences. So N50 ranges from half a kb to 7 kb. Those are relatively short. But what you can notice is that Unitix have most of the genome fraction and the least amount of misassemblies. So we did not submit those Unitix, even though, I mean, I could have submitted them, but I still wanted to provide the decent assemblies. It was just Minia pipeline with tip clipping. So it's one step above Unitix, which has lower genome fraction and more misassemblies, but higher contiguity. And most assemblers performed all simplifications, so look a little bit more like the third line. And I would note that those results are done with end padding. So by considering all contigs, in the assembly in order to have the highest genome fraction as possible. Some quick notes about more detailed steps. So that was a multiple K framework with K up to 91. There was no scaffolding performed. And for the high training dataset, I observed that merging paired and reads didn't always improve genome fraction. That was a little bit counterintuitive, but that was what I observed while doing these tests. And finally, if you're wondering how it performs in terms of computational resources, so Minia used roughly 5 gigs of memory and 5 hours per gigabases in the final assembly. So regardless of how large the input is, what matters is how large the final assembly is. I see a question. Let me try to answer it. What is in the server spec for 5 hours per gigabyte in assembly? Oh yeah. So we ran this in on something like a 20 cores node. So it was a single software, a single machine with around 20 threads. So you need multi-threading. It doesn't just run in a single thread that quickly, but in the order of 10 threads, 10, 20 threads would be. So this is very ballpark. Don't quote me on the 5 hours part. Exactly 5 hours. It's more or less between, let's say, 4 and maybe 10. My final slide is about a comparison between Minia and other software. If you are wondering how other software are doing for assembly. So this slide is not about Mosaic. It's a slide taken from the CAMI, which is a benchmark of metagenome assemblers that was published last year. So each line in this plot corresponds to assemblers tuned to different parameters. So the first line corresponds to both Minia and competitor MegaHit, run with just K equal to 21, so not multiple K. MegaHit has a mode called Mercy K-mers, which enables to retrieve low abundant schemers into your assembly. Minia doesn't have it, so I just disabled it from MegaHit, and there was also no scaffolding. You can see that both assemblers, MegaHit in gray and Minia in blue, are very similar in terms of genome fraction and contiguity. So the assemblies look the same. Second row in this graph corresponds to multiple K. So if you run it multiple times, then you see that both assemblers also improve more or less in the same way the quality of the assembly. And I wanted to show you what the full MegaHit pipeline was capable of. So if you do it with scaffolding, you see that you increase genome fraction, which is a little bit counterintuitive, but it just happens. And you also increase contiguity. And if you turn on Mercy K-mers, then you see also that genome fraction and contiguity improves. So with Minia, we try to deconstruct a little bit what an assembler does in order to have more flexibility in parameters, but we have not performed the later steps. But still, the results can be informative as demonstrated by this challenge. All right, so I'll conclude by just an observation. So if you want to reconstruct strains, if you want to also evaluate the result, there seems to be a trade-off between wanting high contiguity and also limiting the number of misassemblies and genome fraction that you get. So this raises questions on what assemblies to consider and also the state of the art of strain reconstruction. Some references, if you want to cite and use Minia, so I've written the software GitHub webpage and 3 references where Minia was used, so CAMI paper and 2 method papers where we actually studied the representation of the brain graphs and came up with the Minia software. Okay, I'll wrap up. So the slides are available on our webpage, and I acknowledge a few collaborators who were closely or distantly involved in discussion during this challenge. And again, thanks Michalis for giving me the opportunity to speak today.
No, thank you, Ryan. Thank you for participating, and thank you for sharing the method. As mentioned before, if you have any questions, please either raise your hand or click on Q&A. So I have a few questions, Ryan. So you said that it takes 5 gigs of memory to run. Does its performance improve in terms of speed if you give it more memory?
No, in fact, it doesn't matter how much memory you have free. The speed is limited by the amount of data it has to process. And that's fixed.
Okay. And that's great. I think, yeah, no, but it's great still that, you can run an assembler with 5 gigs of memory. There's some assemblers that require like 60 or 70 gigs of memory just to run.
Yeah,
but
if I could add something, we are roughly maybe 5 times less memory than megahit.
yeah, that's amazing. Also, does scaffolding increase the memory requirements?
This I have not measured, but my intuition would be that once you are at the scaffolding step, there is less data to consider. So that would not be the most intensive step.
I see. Okay. So we have a question here that can it handle long reads? And I'm assuming it's from for PacBio data and stuff like that.
About long reads, we have no support for long reads, and it's an interesting problem to try to study, and I will be working on that later.
Okay, that's great. I think we're seeing from our perspective more and more people including long reads in their studies. So we have a question from a user or a participant, let's say.
Yeah.
How does Minia analytical performance compare with Metaspace on the CAMI benchmarking data?
Yeah, that's a good question. Hi, Joey. So we actually didn't perform so many tests with Metaspace, but the one we performed in CAMI showed that Metaspace is more or less the best assembler you can run on any metagenome, as long as you can run it. So Metaspace is very hungry in terms of memory usage and time. It's getting much better because the team has focused on improving their resource consumption. So my general answer would be, if you have the resources to run Metaspace, you will get a slightly better genome fraction and slightly better amount of less misassemblies and much better contiguity than Minia.
Yeah, and that's the important part. I think one of the reasons we're having these challenges is not only to promote the improvement of the analytical results of that you can do with a tool, but also its feasibility, right? So if you have a tool that requires 200 gigs of memory to run, and it gives you perfect or near to perfect assemblies, and nobody can use it, then how useful is it, right? So I think it's great that you two, Ryan, can work on a normal personal computer.
Yeah,
so
this was the aim of Minia from the beginning. I see that you are playing with the results right now, and I would just if you allow me to rebound quickly on that question.
Yeah.
There's also a different matter of goals. So I think this challenge shows that if you are interested in keeping the strains, you might want to play with some hidden parameters in in Metaspace that allows you to do a little bit less simplifications and keep more strains. I think this is what Sergey was a little bit doing in his submissions. And I guess we took it to the extreme by really keeping everything.
Yeah. Yeah, and again, having already said that, we had to make some choices in terms of how we can make this challenge more streamlined. And of course, if we had requested or if we had judged the NGA 50 for the winner, I'm sure people will behave differently, will participate differently using different options. So having said all that, thank you, Ryan, again, for all our users. We have Minia available on Mosaic. So if you go to the tool section, you'll be able to go to the public tools, and you'll see Minia assembler there. If you click on that, you'll be able to run the application. I've already run the results the analysis on some data from strains 2 or the clinical strain detection. And Ryan, it only took like 14 minutes to analyze about 20 million reads, 60 million reads, so that's pretty fast as well. But if anybody of you wants to modify the tool, you can fork it, and then you can change how it works. So I will not spend more time on this, but if you have an account on Mosaic, it's available to you. So thank you, Ryan. So we'll move to the, so we don't have any more questions, I think. So we'll move to the second part of the presentation, which is presentation by CosmosID. And I'll just mention here quickly that for the profiling part, what we evaluated is the cumulative F1 score, which is a measure of precision and recall. And CosmosID performed the best. Again, the way the challenge was constructed is a little bit different than a normal kind of analysis on a microbiome sample. The F1 score here is not exactly the same F1 score you will see in a traditional kind of analysis, and the reason for that is because we're combining the precision recall on different samples from within different data sets, right? So this is just a way for us to be able to evaluate the challenge in a streamlined way. However, what you can see here, if you go to Mosaic, is that CosmosID won this challenge because their analysis performed the best in the analysis of the biosample. And this is something I haven't mentioned yet, but the way we calculate the cumulative score is by weighing the different scores from the different data sets. So the biosample F1 score got the highest weight at 0.4, and the high complexity data set gets 0.3, and so on, right? So, and what you can clearly see here, when you compare the submission by CosmosID to the second best performing submission by an anonymous user, is that in the biosample, CosmosID performed way better than the second submission. If you look at the simulated data sets, you will see that these tools perform similarly, which to me, I think this makes a very important point, is that you know, when you're trying to implement these tools in a clinical environment or in a clinical study, you're always going to be analyzing biological samples, right? So what I think we should be doing as a community is creating more of these real biological samples so we can evaluate and improve the performance of tools, you know, on these biological samples instead of just simulated data sets. And again, you can go to the platform to explore these results. Everything is openly available to you. You'll be able to click on the profiling in the challenge submission area, and then you will see how these different submissions compare. And again, answering participants' question for the profiling part, we have 8 submissions. And again, you'll be able to perform pairwise comparisons, comparisons between the submissions, but also you can dig in the specific submissions. For example, this is a winning submission from CosmosID, and you will be able to dive in and dig in their submission. So let's give it a second. So we provide for you the chrono chart that you can visualize all the different data sets and how they look, you know, stack bars, precision recall charts, and so on. Okay, so with that, I'll turn it on to Hassan from CosmosID. Let me pull up your presentation. If I can find it.
Thank you, Michaelis, and definitely thanks goes to Mosaic platform, DNA Access, as well as Johnson Microbiome, Human Microbiome Institute to organizing these challenges, much needed one. So my name is Nur Hassan. I'm the Chief Scientific Officer at CosmosID. So basically here we're gonna show you a little bit of insight about the CosmosID platform that can be used for cross-disciplinary microbiome research and how we analyze the Mosaic challenge data. Okay, so we all are aware about that huge optimism that microbiome science is exerting today because of its tight interplay between its the host and also in the environment. Every day, numerous publications are coming up with the enormous implication of microbiome with a variety of health and wellness conditions. So, however, with these huge opportunity, it comes up with a lot of unique challenges as well, because the microbiome itself and its host and the environment surrounding it is very, very complex. Therefore, you really need a very highly accurate profiling algorithm, as well as best practices for sequencing production if you want to achieve substantiate a health claim, or if you want to reach your clinical endpoints. To this end, to be successful, definitely 2 things are very, very critical. First of all, you need a very robust analytics that can provide very accurate, complete, and unbiased identifications of microbial community with strain level resolutions, and also can predict the functional potential of the community. And that can be achieved with a robust or class-leading bioinformatics. However, it needs to be actually associated with a large enough sample size so that you can assign some good statistics on the findings. And to do that, it needs to be also a reasonable cost. So to this end, we have actually at CosmosID put our focus on two different things. One is the NGS laboratory services to produce end-to-end microbiome and isolate sequence and analysis using industry standard methods, as well as we focused our bioinformatics development to provide a multi-kingdom microbiome analysis for accurate and actionable microbiome profiling. And with a very good and robust abundance prediction, as well as ability to characterize the functional and antibiotic resistance and virulence potential of the microbiome. So far, our tools has proven to be a class-leading sensitivity and specificity, and it really analyze microbiome at ultra-fast speed. Most of the time, you can process your analysis within a few minutes. So now, as Michalis earlier mentioned that why strain level is important, I wanted to add another information that we most frequently cite, a classical example of Lactobacillus casei. As you know that in a clinical microbiology, your most critical unit for microbial taxonomy is actually strain, not a species, because each strain can confer distinct biological consequences. Here you can see a classical example of Lactobacillus casei, where one strain can be used to produce cheddar cheese, whereas another strain of the same species are used for Chardonnay. On the other hand, another strain has been used as an immunostimulant, which is a component of the DAN active. Similarly, if you think about another most common organisms in a lot of microbiome studies, particularly for human gut microbiome, is the E. coli. So two different strains, like some of the E. coli are beneficial, produce very essential things like vitamin K and vitamin B12 is a beneficial organism to have. On the other hand, if you ever encountered with a strain like O157H7, which causes hemorrhagic enteritis, can make you sick, and very different biological consequence. Therefore, this highlights the need for a profiling methods to be able to identify and differentially detect the strains of biological importance. And now we know that it is so important. Now, how we achieve strain level resolution. So in our platform, we focused on two distinct properties. One is the curated phylogenetically organized microbial genome databases that gives us a deeper dive to understand the gamma diversity of strain populations that can be present in a biological sample. In addition to that, we also have an efficient algorithm, and we use machine learning algorithm to profile across tens of thousands of samples that we have analyzed so far. Combination of these phylogenetic organized comprehensive database and overlay that with this machine learning algorithm to set a very tight threshold to identify accurate prediction of community as well as reduced false positive actually help us attain strain level resolutions with highest accuracy and precision. So now, little bit details about the algorithm and the database. As you know, that CosmosID algorithm is a KMR-based algorithm. So traditionally, most of the algorithm actually organize their database based on phylogenetic taxonomic hierarchy. However, we have taken an approach to organize our database as a phylogenetic tree. So, and once the tree has been constructed, think about 150,000 genomes has been used to create a giant phylogenetic tree, like as a tree of life. And then our algorithm goes and finds KMR signature at every branch and nodes of the tree. And typically, we ended up with hundreds of millions of biomarkers at a different phylogenetic node level. And these phylogenetic tree has been actually painted with the taxonomic informations. Therefore, these organizations help us to identify and differentially detect the closely related strains based on the unique biomarker each of the taxonomic units can contain. Another thing is that our database has been constructed in a way that it is comprehensive, including a lot of different type of organisms, as well as it has database that help you to characterize the microbiome profile. Like if you see that here is a multi-kingdom microbiome profiling that you can achieve based on CosmosID platform. Most of the tools actually looked into bacteria and archaea, whereas we provide both bacteria and archaea community profiling, as well as virus and bacteriophages, along with a lot of micro eukaryotes that has a tremendous importance in a non-bacterial microbiome, and understanding how non-bacterial microbiome contributes to varieties of health and wellness, and also in development of therapy and also for diagnostics importance. In addition to that, it is actually important to understand the community resistance, our effect of our consequences of antibiotic treatment that derived from resistant superbugs, as well as to know how the virulence potential of a community look like. Later on, I will show some example that how you can use all these information to interpret not only strain level identification of the community, but also to understand how each of these can be taken together to interpret that a much more meaningful way. So the second part of our strength, as I mentioned, beside database, is a machine learning algorithm that we used to filter parameters to basically to reduce the false positives and increase the detection specificity. So we have analyzed thousands of different type of samples as listed here, and that gives us to use a balanced way of harmonizing accuracy and precision together, increase the specificity of detection without compromising much of the sensitivity, which majority of the tools typically doesn't include. So this tool you can access. It is open for everybody to access through app.cosmosid.com. Has variety of visualizations and features, and with a partnership with the DNA Nexus, we are offering that for clinical users or clinical trials using like secured and highly compliant and reproducible platform. So now, what is the results of these extensive database with phylogenetically organized structure as well as machine learning algorithm? So you can see it that this is one example that this offers you best-in-class performance. This is a study from where the data sets we analyzed based on the method developed by a group of Cornell researchers. We have profiled those data set based on our algorithm and compared all the performance they have published against into the paper. And as you can see, we definitely shows a unrivaled recall and specificity at a species level. There is a very little sacrifice of precision and recall. However, it become much more prominent compared to the other tool when you see subspecies level. That many of the tools actually cannot achieve to that level of harmonized F1 and precision and recall values. And we definitely are continuously actually benchmarking our tool with all available data sets that is out there. And as our commitment to that, we participated with Mosaic challenges. And in strain 1 challenges, as Michalis showed, that we won the challenges with highest score of strain level microbiome analysis. And most importantly, in the biological sample, which most of the time a researcher will encounter with unknown strain diversity, and we scored 64% better than the second highest submission. So now what we did, how we submitted our results. Basically, when we see the challenges based on the description of the challenges, we first started with our big phylogenetically organized database with 150,000 genome and gene sequences. However, in addition to that, as many other tools has actually started doing, using a small boutique database of with the 440 genomes that the challenges provided, we actually added that one as well. So these are the two submissions we submitted as part of the strain 1 challenge. As you can see in the training data set, that data set, the big gene book database with 150,000 did not perform as well, like as good as the Mosaic database, right? Because it is, most of them are in silico. It does not represent the true biological diversity. However, when we we benchmark, or we actually analyze the data sets of the challenge, as you can see that the CosmosID gene book database actually outperformed the Mosaic database as well as all other tools. So that actually reflects the importance of using extensively curated and a comprehensive database, basically to cover the gamma diversity of the strain subpopulation you can encounter in any biological samples. So this is exactly what Mikhail shows, so I'm not gonna explain much, just to let you know that the first one that was we have two submissions, and both of them actually outperform all other submission, and the top scored one was our big databases, and second one was the boutique databases, particularly created based on the genome that is given by the Mosaic challenge. So we have also have an opportunity to share another news that there is another challenge run by actually DNA Access and FDA, CIFSEN, it's called Precision FDA, and with many other tools, we also have participated, and we are one of the top strain profiling algorithm has been selected, and also we showed the lowest false positive results. Some additional credential, we have been working with the varieties of partners and collaborators in the industries. And basically what we are trying to do, use this microbiome platform in a way to satisfy the needs and applications of these four major verticals, like using this microbiome-centric diagnostic for clinical use, as well as microbiome-centric tool or approaches for microbiome explorations, a lot of microbiome research projects, as well as we have been published and also using these microbiome-based tools for screening and public health applications, as well as a lot of pharmaceutical discovery. And you will find the full list of our publications at app.cosmosid/publications. So with that said, I will touch only a couple of examples in the next couple of minutes. One example I would like to show that that strain interactions in mixed infections, how it works. So this paper has been published in PNAS a couple of years ago. This is an important cases where naturally occurring mixed strain, which typically we consider as a monomicrobial, but here from a single patient, we recovered 4 different strains, and 3 of them are highly virulent, and 1 of them are less virulent. Now the question was this patient has actually lost both her arms and legs and actually died. So in a retrospective study, it was very interesting to see that how these strains actually interact. So with the collaboration with Dr. Ashok Chopra at UTMB, as we did a mouse model experiment, that all 4 strains were mixed together and injected into the muscle. And then we followed up to dissemination of these strain into the peripheral organ. Because we can identify and differentially detect a closely related strain, we created the database along with all these 4 strains. And to our surprise, we see that in the muscle, we are able to recover all of the strains. However, when it's disseminated to the peripheral organ, only the less virulent strain, which is this one, disseminated, which is a big surprise to us because we expected the highly virulent strain would do that. So therefore, we have to validate the results in a laboratory experiment. And so NF1 was actually reconstructed with kanamycin cassette and luciferase gene. And redoing the experiment, as you can see here, you can see that in the mast cell, you see both NF1 and NF2. However, if you would look into the kanamycin plate and also in the spleen and liver, only thing we find is the NF1. So that reaffirmed that actually NF1 is the organism that disseminates to the peripheral organ. That raises a question that how multiple strains interact and in a polymicrobial infection model and help us to identify EXO-A, which is a virulence factor, and also these help us to identify how different strain interact in mixed infection. When you do an avirulent strain alone, it is actually knocked out by the intact local tissue barrier, so nothing happened. If only virulent strain is attacked, then you can see that because of this virulence factor EXO-A, it was able to transmit to the local tissue barrier and disseminate to the peripheral organ. However, when you have a mixed infection, what happened, these EXO-A from the virulent strain actually disrupt the local tissue barrier and pave the path for non-avirulent strain to disseminate to the peripheral organ. So such synergism of virulent and avirulent strain is very important, particularly when in a natural infection, when we try to develop a therapeutic approach, or even also for the diagnostics and proper management of a disease. Next example is the distinguishing pathogens for common cells. We just published a paper with scientific reports where we showed that a lot of blow files, and in the different part of the blow files are actually are carrying Helicobacter pylori, which is a public health threat for many different populations. And most important thing I would like to highlight that not only identification of organism is important, but if you look into the specific virulence factor that these organisms carry, it can give you additional information about the enhanced virulence capability of this organism. For example, here you can see that we find KEG virulence genes and also flagella and FAR virulent gene, which is all are associated with enhanced virulence of this Helicobacter pylori and help to differentiate common cell from the pathogenic organism. The last but not the least one is the consequence of antibiotic use on the resistance and microbiome. We can use the same platform to identify the microbiome signature or biomarker that is associated with different disease state. For example, here we have looked into that what happened in a normal gut when there is no antibiotic. You see a variety of organisms in a pig model. On the other hand, when you treat treat a pig with antibiotic, you see that severe dysbiosis happen, and that give rise to overabundance and underabundance of certain population, which is known to cause a secondary infection. So, and also when you use a drug like SYN-004 developed by synthetic biologics, it happens to protect the deleterious effect of antibiotic in preventing the secondary infection. Similarly, I want to show another things is that when you treat an antibiotics in a piglet model, as you can see, antibiotic actually cause depletion of a lot of common cell normal community in the gut, and simultaneously it also promote the overgrowth of other organisms that may not be beneficial for the host. Not only that, not only the shift on microbiome, but we have also recorded that this also causes the emergence of antibiotic resistance gene. For example, we have particularly see that use of ceftriaxone and actually give rise to enhance emergence of aminoglycoside resistance and tetracycline resistance genes. So with that said, I would say that this type of platform can be used not only for precise strain level identification, but also to understand clinically meaningful information for therapeutic and discovery. With that said, thanks for giving the opportunity. You can definitely, we encourage you to try us at app.cosmosid.com.
Thank you, Hassan. Very interesting. I've learned some things that I didn't know. We have a couple of questions. How does this collaboration with academic groups work? Do you get paid for your services, or is it like a scientific collaboration?
You know, we are a commercial company, right? So most of the cases we do pay for services. However, we have some grant proposal that is with a lot of academic collaborators, and those are paid through the grant mechanism. but all of our work is pay for services.
Okay, we have another question. Do you provide other services like assembly?
We do actually. That is not our cloud approach. This is our in-house, which is very soon we are launching it to the cloud. Right now, we offer all those assembly services, and we actually have another product that is coming soon, and you probably have seen through different webinar and our online communications that we are launching a molecular epidemiological subtyping scheme as well as functional analysis. So these are all coming to the cloud very soon, but right now we are offering all those services through in-house bioinformatics service.
All right, I have a question for you. So you showed the results of your participation in that Mason paper that did the benchmarking?
Yes.
So that was not published, right?
So as I mentioned you that in the original paper, CosmosID was not a tool there. So what we have, we use their methods and we profiled the 34 data sets that they published, and we compared our performance across all the other tools they published there. So that is a CosmosID independent verifications of all these results. And you can see the complete results of these. We write a blog post at CosmosID website. If you go to cosmosid.com and look for the blog, and you can see the entire description of how it has been done and the detailed results there.
Okay, great. So I have a question that is not for you, it's for us. It's about the standard challenge and whether we ship to Europe. And yes, for the standard challenge, you will be able to receive the samples wherever you want in the world, as long as it's legal to do so. So please participate in the standard. Another question. Come again?
Yeah, because there is a question for CosmosID.
Yeah, let me ask it, and you will be able to respond to it. The question is CosmosID genome database available separately on payment to be used with any other pipeline?
At present, no, because our database and algorithm actually cannot be separated. So the whole platform is designed in a way that you use our algorithm to go with our phylogenetically organized database.
I have a follow-up to that, Hassan. So you said it's $99 for the analysis. That includes sequencing and analysis, right?
Yes.
What if somebody comes with their own data?
Yeah, that's fine. So we have two options. Is predominantly, as I earlier mentioned, we have a bioinformatics service, right? You can use our cloud platform to do it yourself bioinformatics. And in many cases, if you need to large scale analysis and sequencing to do it one-stop solution, we do the sequencing service as well. So if you have your own data, you are most welcome to use our cloud platform or contact with us to do a tailored analysis if you need. And if you want standard analysis, I will suggest to buy some credits at app.cosmosid and run your data sets there directly.
And can you share the cost for those analysis, or is it based on case-to-case scenario?
The cost actually ranges based on the number of samples you analyze at once. So it started from it's about $50 to $100 per sample, and basically it depends on the volume, how many samples you are analyzing or how many data sets you are analyzing at once.
So based on that answer, as the cost for analyzing environmental samples the same as $99, or is that only for human microbiome data?
So if you see that $99 price is a kind of a low depth sequencing, it's a promotional for a large quantity of sample. And it includes every type of sample, as long as an end user, if you are looking for a broader overview of community profile, and that will do with $99. And there is a restriction that you have to send samples in a batch.
Okay, great. So I don't think we have any more questions, and we're already 10 minutes beyond our time. So I would like to thank everybody for joining, and especially Ryan and Hassan for presenting their work. This webinar will be posted on mosaic.com. And if you have any questions, please feel free to directly send them to me at mihalis@dnx.com or info@mosaicbiome.com. So with that, thank you all for participating.
Thank you, everyone.
Thank you. Bye.
Bye.